When DNA is found in the cell, most of the time, as the cell is just growing and hanging around, it's not in its condensed form. And you guys are used to seeing DNA in the cell in this X-shaped chromosome. Most of the time, though, it's laying around in what we call chromatin, which is uncondensed DNA, which is just the DNA molecules and its associated proteins. All right? And so this is just a glob of DNA. All right? And so this is what mostly we see. Now, if I were to take the DNA and condense it into what you would see, this is what you would see. One molecule of DNA. This would be a chromosome. All chromosomes must have several things. A linear chromosome, eukaryotic chromosome, has telomeres. It'll have multiple origins of replication located all along the chromosome. And it'll have an area known as the centromere. Now, the centromere can be located anywhere along the chromosome. Ours happens to be here. And this is a condensed area of DNA, all right, to which the kinetochore will be attached. The kinetochore is very important because it's going to be necessary for cell division. This is unreplicated DNA. And so you never see a piece of unreplicated DNA condensed like this. Because as you all know, when DNA is condensed like this into the chromosome form, that happens as the cell is getting ready to undergo replication. And so normally when we see the replicated chromosome, that DNA molecule has already replicated. So we have two strands of DNA attached at the centromere. One strand of DNA, a second strand of DNA. And so that leads to our first question. Is this diploid or haploid? Now, if any of you answered that question, you're wrong, because this has nothing to do with diploid or haploid. Diploid or haploid has to do with the cell itself, and it has to do with whether or not the cell contains a set of chromosomes that come from the mother and a set of chromosomes that comes from the father. So we're talking about sets of chromosomes. A haploid cell contains one set of chromosomes. A diploid cell contains two sets of chromosomes. And that's what determines whether something is haploid or diploid. In this case, this is a chromosome that has been duplicated. Nothing more, nothing less. Two strands of DNA. Okay, now we see that our chromosome has been joined by its friend. This is this chromosome's homologous pair. Alright, so now what we see is we have two chromosomes, same size, same shape. And this is homologs. So let's make sure that we understand a few definitions. This chromosome, these are sister chromatids for this chromosome. These are sister chromatids for this chromosome. These are non-sister chromatids. These are homologous pairs. If I were to be talking about a gene, so for example, if we were talking about fruit flies and we were talking about a gene for vestigial wings, then I might be talking about one fly might be heterozygous. Heterozygous, as you all know, means two different alleles. So on this chromosome, you might have the dominant allele for vestigial wings. And that would be here, located here. Then on the exact same loci over here is where you would find the recessive trait for vestigial wings. All right? Remember, since we're talking about homologous pairs, then this organism that contained these chromosomes would indeed be a diploid organism. Okay, we have a cell, an animal cell here, and it is in interphase. Getting ready for mitosis, we've replicated our DNA, and so all of the double-stranded DNA has now replicated. So there are 2 molecules of DNA in each chromosome. The centrosomes have also replicated, so we have 2 centrioles. And so one of the things that's going to happen is that the centrioles, getting ready to go into mitosis, we're now just going to start it. And the first phase of mitosis is prophase. So the centrosomes and the centrioles are going to start migrating to the poles, which are here and here. The DNA is going to start to condense. As prophase goes along, we start to see that the chromosomes now are beginning to condense, and they're not quite as condensed as they're going to be by the very end. But you can see that they're condensed. The other thing that you start to see is the dissolution of the nuclear membrane. We also see the spindle fiber beginning to form. Now the nucleolus also has begun to disappear. And we are going to be moving into prometaphase. Prometaphase is when the nuclear envelope will completely break down and the spindle fibers will start to connect with the kinetochores. Okay, we're coming into the end of prometaphase. Prometaphase will be characterized by 2 things. All of the nuclear membrane will be completely broken down, and the spindle fibers will be attached to the kinetochores on all of the chromosomes. And when that happens, we will be into metaphase. And metaphase is characterized by one thing and one thing only, and that is all of the chromosomes will be aligned on the metaphase plate. Okay, we are in metaphase now, and the chromosomes are all aligned on the metaphase plate, as you can clearly see. And now we are about to enter anaphase. And anaphase is when the kinetophores are going to divide. And when that happens, the chromosomes, the sister chromatids, are going to move to their individual planes. And we'll see what happens if that works. Hopefully, we'll see what happens. Look at that. And they start moving to their individual planes. Now, as this happens, some very important things to notice. All right? We have individual chromosomes now. So this cell has not yet divided. Right now, you have one cell with 12 chromosomes. 12 individual pieces of DNA for this very short period of time. So anaphase has ended and telophase has begun. And as you can see, the cell membrane has started to pinch in, and we are beginning something known as cytokinesis, and that's going to be the division of the cytoplasm. As telophase begins, we see the nuclear membrane is starting to reform, and the other thing that's going to happen is our chromosomes are going to start to decondense. As telophase ends and cytokinesis is done, we end up with two cells. These are daughter cells. They are exactly the same as the parent cell that we started out with. If we started with a diploid cell, we end up with the diploid cell. If we started with a haploid cell, we end up with a haploid cell. That's the beauty of mitosis.